In this video, we're going to learn about Mendelian genetics, and we'll learn about the patterns of inheritance with simple crosses, monohybrid traits they're called. So first of all, we'll go over what genes and chromosomes are again. So remember, the gene is a unit of inheritance. So it could code for the building of a protein, and that protein could determine your eye color, or that protein could be insulin, for instance. A chromosome is a bundle of DNA that contains many genes. So in this example here, we have a chromosome that has 3 genes, A, B, and C. Chromosomes have more than 3 genes, but for this example, we'll just say it has 3. An allele is they're different versions of the same gene. So when we simplify eye color and we say that people are born with brown eyes and blue eyes, it's a little bit more complicated than that. But in our examples, we'll just say that this is the case. You can have the allele for brown eyes, or you could have the allele for blue eyes. And you can see that one has a capital letter and one has a lowercase letter. We're going to talk about that in the next little bit. So this is an old analogy, but it still works. So back when I was a kid, we used to listen to music on cassette tapes. So a cassette is a large bundle of tape that contains many tracks or songs. So a cassette is like a chromosome. One song or track on the tape is like a gene. The tape itself is used to record the information. So we would say that's like the DNA. All the tape is the DNA. One song on the tape is a gene, and then different versions of the same song, so maybe one person covered it one way and another band covered it a different way, those are like alleles. It's a gene, so the same song, but performed slightly differently, so they're alleles. So different versions of the same song are like alleles. So your phenotype is your physical appearance. So always think about a pH for physical appearance. So that would be brown eyes or blue eyes or red flowers and white flowers. So it's how we describe what the gene is presenting as, what it looks like. Your genotype is the combination of alleles that you carry for a particular gene. So in this example, you could be big B, that's your genotype, or big B, or little b. Remember again that you get 2 copies of every gene because you get one from your mom and one from your dad. So you can have 2 of the same allele, or you could have 2 different alleles. It all depends on what you inherit from your parents. So a dominant allele is an allele that's always expressed when it's present. So it can't be masked by another one. So a dominant allele, you only need 1 copy. So you could be big B, or big B to have brown eyes. You can't mask it with another allele. You only need 1 copy. A recessive allele is an allele that can be masked by another allele and is only seen when you have both copies of the recessive allele. So little b will give you blue eyes. So there's a few more terms to be aware of. So if an individual has 2 copies of the same allele of genes on a pair of chromosomes, the individual is homozygous. So in the example that we've been using so far, you can be homozygous if you're big B, or little b. Homo means the same. So you have 2 of the same alleles. Another term sometimes when you're reading examples and problems is called purebred. So you're pure breeding. You have 2 of the same allele. If an individual has 2 different alleles of a gene on a pair of chromosomes, they're called heterozygous. And hetero means other. So big B, little b would be an example of a heterozygous individual. That is also called hybrid because they have a blending of the 2 alleles. So in biology, we use something called a Punnett square to work out the probability of the different genotypes and phenotypes in a genetic cross. So in step 1, you would write down the genotypes of the parents, and in step 2, you would write the genotypes of the gametes that each parent produces, and then you would draw your Punnett square and figure out the probability. In this example, we're going to do a cross between two red flowers that are heterozygous. So we'll imagine that red, the dominant allele is red, so big R, and the little r will be for white, the recessive allele. So the two parents are heterozygous for both, so we're crossing that as big R, little r, being crossed with another heterozygous big R, little r. So we need to write down the genotypes of the gametes that each parent produces. So each parent could give a big R to the offspring or a little r. So that's parent 1, and parent 2 is the same thing. So we could give a big R or a little r. So those are the genotypes of the gametes. So the next thing is we draw our Punnett square. This one's drawn a little bit more complicated. I often just draw a box and then just put the genotypes above and below. But we draw the genotype of the different gametes in your Punnett square. So big R, little r for the eggs, big R, little r for the pollen, and then we match them up. So big R matches with big R. We have big R going with little r, big R with little r, and then two little r's. So again, you could have just done it simply like this without labeling it eggs and pollen. So again, same thing, big R, big R, big R, little r, big R, little r, little r, little r. We always write the dominant allele first, so that's why I didn't write little r, big R for the last one. And then this will show you the genotype possibilities for all the offspring. So those are the odds of what the outcomes are going to be. So now we'll take a look at the probability of the genotypes. So that's the combination of genes. So there are 4 possible ways to combine egg cells and pollen. So you count up how many times each genotype appears in the Punnett square, and that gives the probability. So only one time can we ever have a big R, big R. So the probability of big R is 1 out of 4, or 25% of the time. There are 2 options for big R, little r. So that's 2 out of 4, or 1/2, or 50%. And the probability of little r is also 1 out of 4, or 25%. And if we were to write that out as a ratio of big R to big R, little r to little r, it would be 1 to 2 to 1. Now, if we look at the genotypes, we can decide the phenotypes each one should produce. So if you take a look, there are 3 options where you end up with at least one big R, and that's the dominant allele. So we've got this one, we have this one, and we have this one. So the probability of red is 3 out of 4, or 75% of the time you should have a red flower. The probability of white is just 1 out of 4, or 25%. So the ratio of red to white is 3 to 1, and that's how you do the notation.